So our next speaker is Dr. Sandra Dross from the Dr. Fuller's laboratory at the University of Washington, and also she's a recipient of our scholarship this year, and so she was selected as from the abstracts submitted. Congratulations.
Hello. So I will start with my community summary. So we think that cure will likely require not just one treatment, but a combination of treatments in order to deal with some of the complexities of HIV, including the rapid mutation rate, the exhaustion, and persistence of latent virus. One of our key questions was whether using a combination of treatments in a non-human primate model would work together to overcome these issues to cure infection. So here, this is based on a comic, The Oatmeal, which is a local Seattle comic, and I use this to represent our combination of treatments. So we have anti-PD1 to revive exhausted T cells, DNA vaccination in order to target T cells towards regions that are really critical for viral survival, GS986 to awaken infected cells that are then seen and destroyed by the immune system, and CCR5 editing to protect CD4 T cells from reinfection. So one of our key findings was a little disappointing. We didn't see during stopping ART that there was a significant decrease in the viral loads of our treatment versus control animals, but animals with lower viral loads had higher specific vaccine responses, and we think that we can induce changes in our further studies to improve our combinatorial therapies and our vaccine responses. So the take-home message is that our vaccination is working to induce conserved element responses, and we need to improve our combination of treatments to achieve cure. So within our Collaboratory, we are involved in IRF3, which is using a combinatorial intervention to either reduce or eliminate the viral reservoir in HIV-infected rhesus macaques. So we are using a conserved element DNA vaccination. We're doing this to target the immune response towards conserved regions of virus in GAG and ONV. We give this DNA vaccination 5 times. We are also using exhaustion reversal via anti-PD1 therapy. We give the anti-PD1 therapy prior to the first, third, and final DNA vaccinations. This is to enhance our vaccine immune responses. We are giving GS986 to reverse latency. This is a TLR7 agonist, which should activate our infected T cells. And finally, we have CCR5 editing to protect CD4s from reinfection. We are using a zinc finger nuclease to do this. So we have a total of 24 HIV-infected animals in this study. We have 8 animals per group. So we have our control group in blue, our vaccine and CCR5 editing group in green, and our combinatorial group receiving all of the interventions in orange. And these colors will remain the same throughout the presentation. So I'll sequentially walk through the immunological outcomes of our interventions, starting with the DNA vaccination and anti-PD1 therapy. So we found if you look either before vaccination and intervention versus after the 4th vaccination, we found a significant increase in the T cells, both the CD4 and the CD8 T cells in the peripheral blood on the left and the mesenteric lymph node on the right in the 16 vaccinated animals. These animals had not yet received the GS986 or the CCR5, so right now the differences between the 2 groups are simply receipt of vaccine plus or minus anti-PD1, and I'll show that in the next graph. A little bit surprisingly, we found a differential effect of the anti-PD1 therapy prior to vaccination. So in the peripheral blood, we saw that the magnitude of these T cell responses were higher in the combinatorial group receiving anti-PD1. However, in the mesenteric lymph node, we saw that the magnitude of T cell responses were higher actually in the vaccine-only group. This suggests to us that anti-PD1 may not traffic to the mesenteric lymph node, or it may induce some kind of a differential response there. I'm not going to show this data in the interest of time, but these were our peak vaccine responses. They waned prior to ATI, and we're working to resolve this issue. So next, I'll talk about the GS986 immune response. So A little bit differently compared to the original GS986 paper by Whitney et al., we did not see any viral blips. We saw one viral blip in 8 of the animals in 10 of the GS986 doses. We additionally did not see CD4 or CD8 T cell activation or plasmacytoid dendritic cell activation. This is in agreement with Jeffrey Lifson's most recent paper showing a similar finding, suggesting that our GS986 wasn't really adequately reversing latency in these animals. And we're additionally looking at the plasma cytokines, as Ramaz has suggested yesterday during the SAB meeting, to validate this. So lastly, I'll talk about the CCR5 editing protocol. So in our control animals versus our treatment animals, we did not see an impact of the CCR5 editing protocol on our CCR5 expression in the peripheral blood. Paradoxically, we actually saw a significant increase post-infusion in the animals that are CCR5 edited. After discussions with the group, we think this is stochastic and probably related to the fact that these animals are undergoing ATI at the same time. Regardless, we did not see an impact or a reduction of CCR5 expression. So finally, I'll show the viral load kinetics post-ART in our controls versus our treated animals. So up top, I'm showing the control animals' viral loads either before the treatment interventions and then after during ATI. In the middle is the vaccine and CCR5 group, and then in the bottom is the combinatorial group. And as you can see, we did not see a significant difference in the ATI viral loads. We didn't see a difference in the peak viral loads, the set point viral loads, or the area under the curve viral loads in these animals, which is a little bit disappointing. But we did find some significant correlations between specific vaccine responses in the mesenteric lymph node and ATI viral burden, suggesting that these responses may be important for viral control. This agrees with a previous publication in our group showing that gut-associated vaccine responses and not peripheral responses are important for viral control. So we're interested in following this up in the future to enhance our vaccine efficacy. So in summary, we found that CE vaccination enhanced CE responses, but these did not persist, and we saw a differential effect of the anti-PD1 dosing. We will be, in the future, determining if trafficking of anti-PD1 or the effect on exhaustion in the blood or tissues is different. And we have an upcoming pilot which will utilize a novel adjuvant in order to enhance persistence and mucosal homing, as we found that those responses were important for viral control. We found that GS986 did not induce viral blips or cellular activation, and so our Collaboratory is looking into utilizing alternate latency reversal agents. Our CCR5 gene editing did not reduce our CCR5 targets for infection, but we think that this is a rather easy fix. The plan is to infuse a larger number of edited cells to lower the CCR5 percentage in the blood. Our editing efficiency was quite high. The median was 60% of editing efficiency, but the total edited cells in the blood after infusion was 0.1%. So we think that with a larger cell dose that we can actually impact the CCR5 expression. And then finally, we found that mucosal CE responses did correlate with viral burden, which suggests to us that enhancement of these responses may increase viral control during ATI if we are able to modify our combinatorial treatments. So I'd like to thank the Fuller Lab and point out that both Megan and Hillary are giving posters. Please visit them. Thank the UM1 Defeat HIV team and our funding, including the scholarship, and also point out that the Fuller Lab is seeking an exceptional postdoctoral fellow if anyone is interested. So thank you.
Thanks a lot, Sandra. Any questions? There's no questions. There's else?
Yes.
I'm going to shut. How did you introduce the CCR5 editing? I missed that.
Sorry, I was speaking so quickly. It's an oral abstract, so I have to go quickly. So this was via zinc finger nucleases, and it's a similar protocol to Sangamo. It's a rapid production, so we have a 6-day total editing protocol that, and Chris can talk.
Efficiency do you estimate? How much efficiency do you think you have?
We saw a range between 40 and 80% editing efficiency, but then, as I said, the cell dose was quite low, so the overall edited cells in the blood was 0.1%.
Thank you.
Okay. If there's no other questions, I think, thank you very much again, Sandra.